Well, I'm running out of time to put these problems up before, so you'll have enough time to look at them before, our exam on Tuesday. It's, Sunday night right now, about 6:30. So I'm going to try and do these one shot, and it might be kind of messy, but at least I will have them finished. So let's take a look at problem 13.4. The messenger RNA formed from the repeating tetranucleotide UUAC incorporates only 3 amino acids, but the use of UAUC incorporates 4 amino acids. Why? Well, let's take a look at this. So now remember, we had a lecture, and I really like this lecture because it's fun, I think, to see how scientists in the 1960s and early 1970s went from knowing nothing about how the information in DNA and RNA could direct the synthesis of a protein. Can tell the cell how to put one amino acid, a specific amino acid, at one position, followed by a specific one at the next position, followed by a specific one at the next position. Like, how did that happen? Scientists didn't know it, and it's really, I think, neat to see how they discovered these things. And by studying and by learning how scientists did these things in the past, hopefully we can use similar methods and similar thinking to discover new things in the present and future. But I really like the discovery of the genetic code, elucidation of the genetic code, and that's kind of why we spend a lot of time or a lecture figuring out how they did it. Plus, it's just a neat way to get used to working with the genetic code. So, this question we are working with a repeating tetranucleotide, 2 of them. We're going to start with UUAC. So 5' UUAC. Now, we talked about a couple different types of RNA polymers that the scientists really used to decipher the code. They were Homo Polymers, which were essentially just long chains of U's and long chains of A's and C's, and we said G's didn't work, but we don't have to remember that. Then we had copolymers, which were mixtures of nucleotides, and they were using that enzyme, polynucleotide phosphorylase. And specifically, we had random copolymers, where they put in certain ratios of bases with this. They mix certain ratios of the bases with the enzyme, and they're able to get mixtures of copolymers with 2 different nucleotides in there. And because they know the ratios, they can predict how frequently they should encounter certain combinations of bases inside 3 base codons. Like how often they'll see a codon with 2 A's and 1 C, and how often they'll see a codon with, say, 2 C's and 1 A. So that would be the random copolymers. And we also talked about the repeating copolymers. So with this one, this happened a little bit after the development of the random copolymer assay. The repeating copolymers involved taking small pieces of RNA that scientists have devised a way to make small chains of RNA of specific sequences. They couldn't make it specific RNA much longer than 4 nucleotides, but they could make lots of these, thousands of these. And Gobind Khorana figured out that, hey, I can stick these together, one after another, and get a repeating copolymer. Okay, and when we use UUAC, we can see that there aren't that many 3-letter codons in here. So if codon has to be 3 letters, well, we have a UUA here. We have CUU. We have ACU. We have UAC. Okay, then we have UUA again. Oh, wait a second. Okay, we already have that one. How about CUU? We already covered that one. And so I think we'll find that we only see 4 possible codons in here. Remember, codons have to start with a 5' end with a 3'. So UUA here is different from AUU. So those are different. The 5' and 3' ends matter. So we have UUA, we have CUU, we have ACU, and UAC. So there should only be 4 possible codons within this long chain of RNA. So the question is saying UUAC incorporates only 3 amino acids, but UAUC incorporates 4 different amino acids. So apparently when we use this long chain of RNA, we add this, and what did they do back then in the 60s? They added this to a protein-producing cell lysate. And they watched how often or what types of amino acids were incorporated into the protein made by this lysate. And then, when they figured out that, say 25% of the protein was glycine and 25% was alanine, and let's say phenylalanine was the other 25. And let's say there was a 25% was, I don't know, let's come up with another one. Give me another histidine. So they would know these 4 proteins had to correspond with these 4 codons. I didn't look these up, so these probably correspond with other proteins, but that's essentially how it worked. They look at lots of different repeating copolymers, and they're able to decipher the code that way. But the question says, these right here only incorporated 3 different amino acids into a protein. So these 4 correspond to 3 amino acids. And this next one down here, let's see, what was the other repeating unit was UAUC. So the first one was UUAC, this one's UAUC. UAUC, UAUC, so on and so forth. Okay, now let's see how many codons we see here. We have UAU, we have CUA, we have UCU, we have AUC, and then we're repeating again. Okay, so let's write those down. UAU, CUA, UCU, and AUC. I'm going to put the 5' and 3' in here. And so the question now is, okay, we see 4 codons in both, resulting from both repeating copolymers. We can find 4 codons in each one, but for some reason, this repeating copolymer only leads to the incorporation of 3 amino acids, and this one down here leads to the incorporation of 4 different amino acids into the protein. So I haven't looked at the answer, but I'm guessing it's probably going to be because the genetic code is degenerate. Now let's take a look at that. So let's take a look at that. I think the easiest thing for me to do would be to copy. Okay, these are the codons from the first repeating copolymer. I'm going to put those right here. And these right here are the codons from the second repeating copolymer. I'm going to put those over here. Now remember, the question told us that these on the left lead to 3 amino acids being incorporated into protein, and these over here lead to 4, I'm going to check that again, 4 amino acids. So let's see if I can paste the codon table in here. Okay, here's a small codon table. Now let's see what these codons correspond to. So I'm going to look for UUA. So let's see, UUA, let's find this one. UUA, I'm going to start here, first position U, second position U, third position A, so UUA is leucine. So this one codes for leucine. This one right here, CUU, first position, second position, U, leucine. Okay, it's starting to make sense. ACU, where is ACU? Threonine, right here. And let's see, UAC, UAC, tyrosine. Alright, tyrosine. This is Dan's favorite amino acid. He told us that in class. So, let's see. Only 3 different amino acids. Okay, everything's making sense, right? So how can there be 2 codons that both code for leucine? Well, that's because the code is degenerate, right? So more than one codon can code for the same amino acid, and that makes sense because there are 64 possible codons from a 3-letter. When codons are 3 letters long and there's 4 possible bases at each position, that would be 4 times 4 times 4, 64 possible codons, but there's only 20 amino acids that the codons need to code for. So everything's making sense. So my guess is these 4 codons over here are going to code for 4 different amino acids. So let's see, UAU is going to be, this is recording, right? Yes, it is. Okay, so UAU, tyrosine, right here, CUA, leucine, UCU, serine, and AUC, isoleucine. Okay, so this was a fun problem. You had to know a little bit about how repeating copolymers work, how the assay worked. Need to understand a little bit about that, and need to know how to use codon tables to look up codons in the amino acids that they code for.